This will be the third part of the male reproductive physiology presentation, and it will start with spermatogenesis. Spermatogenesis is the process by which haploid spermatozoa develop from germ cells in the seminiferous tubules. This process starts with mitotic division of the stem cells located close to the basement membrane of the tubules. Sperms, each day about 300 million sperm complete spermatogenesis. Each day there are 300 million sperms complete the spermatogenesis. And the sperm has a head, neck, and tail. Head, neck, and tail. The head has an acrosome which produce penetrating enzymes to penetrate the egg, and a mid piece in the neck which contains the large mitochondria to provide energy, and a tail to help the movement forward of the sperms. The function of the sperms is to move and carry genetic information in the DNA in the nucleus. 23 chromosomes carries DNA in the nucleus to the egg. Structures of the sperm. The sperm is 60 micrometer long. It has head with the acrosome, and the acrosome secreting oocyte penetrating enzymes. The start of the spermatogenesis start in the seminiferous tubules, in spermatogonia, spermatogenic cells, which is the sperm forming cells, spermatogonia, stem cells, develop from primordial germ cells coming from the yolk sac to enter the testes in the fifth week of development. Primary spermatocytes, these primordial germ cells change into primary spermatocyte, and spermatocyte change into secondary spermatocytes, to spermatids, and then to sperm cell, then the lumen, then goes to the lumen of the seminiferous tubules. Two cells controls this process: Sertoli cells, support cells. It has tight junction. It prevents immune response against the sperm cell surface antigen, and also provide nutrients to the sperms and secrete inhibin. It nourishes spermatocytes, spermatids, and sperm itself. Provide nourishment for all these cells and control the movement of the spermatogenic cells. It releases sperm into lumen and produce fluid for sperm transport, all this by the Sertoli cells. It secretes inhibin, which regulate the follicle stimulating hormone, and also the Sertoli cells regulate the effect of testosterone. Leydig cells, another type of cells controlling the spermatogenesis. It is in the interstitial cells found in spaces between seminiferous tubules, and these cells secrete testosterone under the control and effect of luteinizing hormone from the anterior pituitary. So these diagrams showing that spermatogonia diploid undergoing mitotic division to form primary spermatocytes with diploid number of the chromosomes. Then these primary spermatocytes will undergo the first meiosis, first meiotic division, to form the secondary spermatocyte with haploid number of the chromosomes, with haploid number of the chromosomes. The secondary spermatocytes with haploid number of the chromosomes perform second meiotic division to form the spermatids, which are the haploid, which contain the haploid number of the chromosomes. Spermatids with two stages of differentiations turn into sperm cells with haploid number of the chromosomes. So the primary spermatocytes perform first meiotic division, and the secondary spermatocytes increase the number of the spermatids by a second meiotic division. And the primary, the spermatogonia with diploid in a continuous meiotic division to form primary spermatocytes with diploid number of the chromosomes. This process under the control of hormones from the hypothalamus, gonadotrophin releasing hormones, stimulate the release of the follicle stimulating hormone and the luteinizing hormone. The follicle stimulating hormone produce second messenger enzymes to stimulate the Sertoli cells to produce fluids, nourishment, support for the spermatids, and also secretes inhibin, which also control the FSH. The luteinizing hormone, under control of the gonadotrophin releasing hormone, stimulates the Leydig cells to secrete testosterone to body for secondary sexual characters, and to Sertoli cells to also stimulate its function. And the testosterone also is important for the whole process of the spermatogenesis. Male sexual act, it has three stages: erection, emission, and ejaculation. These are under nerve control, whether it is parasympathetic, sympathetic, or somatic nerve controls: erection, emission, and ejaculation. The parasympathetic nerve, sacral 2, 3, and 4, supply to the erectile tissue of the penis, take part in the erection of the penis. The sympathetic nerves from thoracic 12, lumbar 1 and 2, supply to the epididymis, vas deferens, seminal vesicles, ejaculatory duct, take place in the emission, which is preparation of the semen. Somatic pudendal nerve supply to the pulpus cavernosa. It takes place in the ejaculation. Emission and ejaculation, again, sympathetic nerve, thoracic 12, lumbar 1 and 2, form the hypogastric and pelvic sympathetic nerve plexus to initiate emission and ejaculation. Emission and ejaculation. Preparation of the semen and ejaculation of the semen. It also leads to contraction of the vas deferens, expulsion of the semen to the urethra, contract prostate and seminal vesicles, expel prostatic and seminal fluid into the urethra. All the fluid mix in the internal urethra with mucus emission and is called for runner for the ejaculation. This under control of the sympathetic nerve, the thoracic 12, lumbar 1 and 2, which form the hypogastric and pelvic sympathetic nerve plexus. Seminal fluid, which is the semen, it is a mixture of seminal plasma and cells. Prostatic fluid, 30% of the volume, and epididymal plasma, 5% of the volume, and seminal vesicle fluid remainder, which is around 60%, 65% of the ejaculate. The cells of this semen are sperms, the spermatozoa, germ cell or germ-line cells, leukocytes, white blood cells, bacteria, epithelial cells, and occasionally red blood cells. Why do we perform semen analysis? We perform semen analysis to diagnose fertility, infertility, sterility, and to identify treatment options, whether it is surgical, medical, or assisted reproduction. Sample collection must have some rules that it should be in a container which is sterile, 3-5 days abstinence, and under hygienic conditions. The sample must be kept at 37 Celsius centigrade degree until analysis, which begins and ideally within 30 minutes of ejaculation. These are the WHO characters of normal semen. The volume of the semen should be above 1.5 ml. Sperm concentration per ml 15 million. Total sperm number at least 39 million. Progressive motility 32%. Total motility 40%. Vitality of the sperms, at least 58% of the sperms are alive, with more than 4% morphologically normal. The pH of sperm of the semen should be less than 7.2. Leukocytes, white blood cells, less than 1. And the immunobead test to check the antibodies against the sperms should be less than 50%. Sorry, the pH of the semen should be more than 7.2. It should be an alkaline, but around 7.2, not more than that. This will be the end of the third presentation, and the next presentation will start with erectile dysfunction. Thank you.